I'll be actually giving two different seminars on the day. The first one is actually going to be in the gene therapy session. We'll be discussing a novel gene that we've discovered and are using as a gene therapy approach for the treatment of several different types of diseases. It's the gene is called Mitsugumin 53. It's also known as TRIM72, and the gene itself is involved in the resealing of membranes after they're disrupted by a variety of different types of insults. So these sort of membrane disruptions of individual cells take place in a lot of different disease states, and when that occurs, it can lead towards the death of the cell. The accumulation of the cell death will lead to the eventual failure of the target organ. We principally focus on this in muscular dystrophy and in cardiovascular disease, targeting the repair of the sarcolemmal membrane, either in muscular dystrophy when it's disrupted by mechanical damage to the membrane, or in heart failure when ischemia-reperfusion injury causes damage to the membrane. And when that occurs, these cells would normally die unless the cell can reseal that damage and allow the cell to survive and the tissue to continue its function. The MG53 protein is important in driving this process and maintaining the operation of the machinery that will reseal the membrane. And if we overexpress that through genetic means, we find that we can treat both the disruption of skeletal muscle damage during muscular dystrophy, and also we can help prevent some of the damage associated with ischemia-reperfusion injury during heart failure or before heart failure. That's the first lecture in the gene therapy section, and we'll be discussing that in a gene therapy sense. And we'll also be discussing in the cardiovascular section. We'll also be talking about some of these uses of MG53 in the prevention of cardiovascular disease, looking principally at overexpression studies and in the gene therapy approach, and also looking at the ways that we can target other ways of doing regenerative medicine in cardiovascular disease. Another program we have in the laboratory focuses on the use of stem cells, coupled with a novel encapsulation technology. It's a microfabrication technique using electrospray that we do in collaboration with another group. And then we test that in various cardiovascular disease models based in rodents, and look to see if we can have regenerative abilities in the damaged heart and whether that's detected fast. So that's the basic background of the two different topics.
So the summit basically focuses on the advances and innovations. So what kind of innovation and advances are you going to talk about in the presentation, sir?
Well, a lot of the innovation focuses around in the case of the stem cell project, the fabrication techniques that can be used to deliver the stem cells. So obviously stem cells are a well-established technology. They haven't reached the clinics yet, but are a well-established experimental technology in regeneration of a lot of different organ types. But there's certainly challenges associated with the use of those technologies. And one of them is the ability to deliver the cells effectively to the target tissue. Another one is allowing those cells to survive once they implant in that tissue. And our preliminary results and that we're pushing forward with this project suggest that our encapsulation technology is capable of improving both those aspects. So that's really the innovation aspect of that. While stem cells are fairly established as an experimental approach, this encapsulation technology is a new way to be able to apply stem cells towards the treatment of cardiovascular disease. And from the point of view of therapy with gene therapy, that's the novel aspect is the use of the MG53 gene. It has a potential utility in a lot of different disease state where there's a great deal of necrosis that takes place that contributes to the pathology of that disease. So we're focusing on the novel nature and the innovation that can be provided by targeting membrane repair as a treatment for disease.
Okay. And what kind of learning should be drawn, sir, from this? Because there would be delegates who would be medical students, and there would be, medical equipment manufacturers, there would be doctors, there would be surgeons. So in total, what should be the learning that these delegates would, drive from the area of presentation that you have chosen, sir? In brief, what should be the learning that they should take home?
Well, I would hope after these lectures they'd have a better understanding of two things, really. The ideas that are related towards membrane repair what the fundamental mechanisms are and how those can be targeted as a therapeutic approach for the treatment of different disease states. So we'd be focusing specifically on muscular dystrophy and cardiovascular disease, but how they could be applied to other disease states as well. And second, it would be one of the goals for them to be able to understand this fabrication technology, what the advantages are, what the needs are in order to be able to scale it to a higher utility, so it could be used in a general manufacturing procedure for different types of pharmaceuticals, and as well as the delivery of stem cells.